Hi, everyone. I'm delighted to be here today to tell you a little bit about the project that I've been working on, which focuses on the development of new metabolic therapies for heart disease and heart failure by harnessing the regenerative potential of the pediatric heart. Now, the heart failure is generally described as the inability of the heart to meet the metabolic demand of the body. And the reason why we're interested in it is because despite the current treatments for the disease and the advancements in knowledge about the disease, the 5-year survival for these patients is still only 50%, really necessitating the development of new effective treatments. Now, oftentimes you might hear a failing heart being referred to as an engine out of fuel, which really highlights the metabolic dysregulations as one of the hallmarks of the disease. And so usually what happens in a normal adult cardiomyocyte is that the majority of the energy comes from the oxidation of fatty acids, and the majority of the rest comes from the oxidation of glucose in the cell. But what happens in a failing heart is that this balance in metabolism is shifted from fatty acid oxidation towards glycolysis. And what I really want to highlight here is the significant difference between the energy yield in a healthy heart versus a failing heart. And what we want to do is to address this metabolic dysfunction in a failing heart by really harnessing the regenerative potential of a pediatric heart. So usually in an adult, unlike an adult heart, what happens in a pediatric heart is that they have the capacity to repair and regenerate themselves after an injury. And much of this capacity comes from these resident progenitor cells in the heart that are called mesenchymal stromal cells. And interestingly, our lab has shown that the pediatric heart or a young heart has a higher abundance of these cardiac progenitor cells. Not only that, but these progenitor cells also secrete a variety of biomolecules into their extracellular environment, collectively known as the cell secretome. And what has been shown is that the secretome that is collected from a neonatal heart has a higher reparative potential compared to an adult. Now, the biomolecules that are released by the cardiac progenitor cells, they can induce cardiac repair through different mechanisms. For example, they can reduce fibroblast activation by modulating the inflammatory response in the cell and limiting the pathological remodeling of the cardiac tissue under disease conditions. They can also enhance proliferation of cardiac progenitor cells and enhance neovascularization in the tissue. But more relevant in the context of heart failure, they can induce cardiomyocyte survival and contractility, which is highly dependent on metabolism. So considering the reparative potential of the pediatric cardiac progenitor cell secretome and the importance of metabolism in cardiomyocyte function, we wanted to know if these secretomes can enhance metabolism, if so, what is the mechanism behind this enhancement? So to answer this question, the first thing we did was to obtain these patient secretomes. So we collected patient tissue, pediatric patient tissue from patients who presented at Sick Kids for open heart surgery. We bring these cardiac explants to the lab and culture them and grow them. And then we collect the conditioned media that contains all the secreted factors that these cells produce into their extracellular environment. And then use that concentrated secretome. So we concentrate it through a series of steps and then use that in our in vivo and in vitro assays. So after obtaining these secretomes, then the first thing we wanted to answer was, are these secretomes active and can they induce a beneficial phenotype in vitro? And to answer this question, we injected these secretomes intramyocardially in a rat model of cardiac injury. And then we followed these rats over 28 days. So these images that you're seeing here, these are echocardiograms of at 28 days of rats that are injected with control media or rats that are injected with the patient secretome. And what you're looking at is movement of the ventricle on the y-axis over time on the x-axis, which with each one of the peaks being the ventricle contracting and the troughs being the ventricles relaxing. And just by looking at the two images, you can see the significant difference between the contractility of the two groups. But to quantify this change or this difference, we looked at two different metrics of cardiac contractility. One, we looked at fractional shortening, which is a direct measure of cardiac contractility. And what we see is that rats that were injected with the patient secretome recover back to baseline by 28 days, showing enhancement in contractility. The other metric that we use is left ventricular end systolic diameter, which, as the name suggests, is the diameter of the left ventricle at the end of a contraction. So the smaller this number is, the better the contractility of the heart. And again, we see that rats that were injected with the patient secretome show better contractility. So now that we know these secretomes that we have are actually active and they induce beneficial phenotype, we then wanted to see, do these secretomes affect metabolism? So to answer this, we treated iPSC cardiomyocytes with these patient secretomes. And then we did this both under normal oxygen conditions and under hypoxia. And what we see is that both under normoxia and hypoxia, and then when we measure different aspects of mitochondrial bioenergetics, we see that cells that were treated with the patient secretomes have a higher basal respiration, mitochondrial ATP production, and maximal respiration, all of which are indicative of a better functioning mitochondria and better mitochondrial activity in general. And what I want you to really appreciate here is that each one of these data points in the patient secretome group, these are not just biological or technical replicates. These are cells that are treated with secretomes collected from different patients. So then, as one potential explanation for this enhanced mitochondrial respiration, we looked at mitochondrial polarization. Sorry, my slide is a little weird looking here, but basically and on the top panel, we use JC1-DI to look at mitochondrial polarization. And JC1-DI, it exists in two forms. And in the top panel, what you're seeing is the aggregate form, which fluoresces in red. And the bottom panel is the monomer that fluoresces in green. And we report mitochondrial polarization as the ratio between aggregate to monomer. And if you look at the last two panels, on the right, so these are the last, the one before the last is these are cells that were treated under hypoxia, or just they were incubated under hypoxia for 24 hours. And the last panel, these are cells that were under hypoxia and that were treated with the secretomes under hypoxia. And what you see is this significant enhancement in mitochondrial polarization. So So far, we know that these cells enhance mitochondrial respiration and they enhance mitochondrial polarization. So moving forward, what we want to determine is how these secretome treatments can alter the use of energy substrates, since different energy substrate can have different ATP yields in the cell. And also we want to verify these in vitro observed mitochondrial changes in vivo in the rat tissue that we have collected previously. And finally, and the most important part of the project is to really to determine the protein constituents of these patient secretomes through mass spectrometry, and then identify potential metabolic and mitochondrial therapeutic agents that are present in these secretomes as potential therapies for heart failure. And with that, I would like to thank my supervisor, everyone in the mains lab, my advisory committee, and all our funding sources.